Alright, so we are looking on the monocot root cross-section. Smilax is our example here. And we're going to start on the outside with the epidermis layer, always on the outside here. And this particular epidermis has a pretty thick cuticle, that waxy layer on the outside. It's kind of a dark red in that image. But all of that is, this layer would be the epidermis. And each one of these cells would then be an epidermal cell. And these two things are part of the dermis tissue. Okay. The next layer, and actually I'm going to come in quite a bit and do this layer in here, because that really distinguishes the region between the two. This very thick dark red area, that is the endodermis. And between these two areas, this whole region would be the cortex. And the cortex consists of these very large cells, dodecahedron, that are loosely packed in here, forming intercellular spaces between them. Okay. So each one of these single cells is a parenchyma. And the triangle shape at the points where they meet, their area is intercellular space. And the parenchyma cells are filled with small organelles called plastids. There's several types of plastids, but in this case, each one of these is an ameloplast. And an ameloplast is a plastid that stores sugars. So that's what's happening here. The one other thing I wanted to mention about this space is that all of these cells and all of this tissue is part of the ground tissue. So the region is the cortex, but the tissue here is ground. All right. The endodermis, as I already mentioned, is this really thick red area. And let's see if I can do it like this way. The thing that makes it red is called the Casparian strip, and we'll talk more about that. But the Casparian strip lines the walls of these cells like mortar in a brick. Okay. And this internal area, everything inside the stele, the endodermis is the stele, and that consists of lots of vascular tissue, xylem, and phloem.